SWOG S1609 DART is the first NCI-funded basket immunotherapy study with over 37 rare tumor cohorts in which patients are receiving ipilimumab plus nivolumab, dual immune checkpoint inhibition. The cohort that was discussed at AACR was the neuroendocrine cohort, in which patients with high-grade neuroendocrine carcinoma had, at a 44% rate, had shrinkage of their cancer versus patients with lower intermediate disease who had a 0% rate of shrinkage of their cancer with these two agents. We're not quite sure why patients with high-grade neuroendocrine carcinoma conferred the vast majority of the benefit in this study. One of the hypotheses is that it relates to tumor mutational burden. And so what we're doing is assessing these tumor specimens through our collaboration with SWOG CMAX in order to better understand the patient's tumor biology and determine whether tumor mutational burden, PD-L1 expression, or another aspect of the patient's unique immune characteristics within the tumor determine whether they would respond to dual immune checkpoint blockade. SWOG S1609 is a master basket protocol in which patients across 37 different rare tumor histologies receive ipilimumab 1 milligram per kilogram every 6 weeks continuously with nivolumab 240 milligrams every 2 weeks. This is a lower dose of ipilimumab than what is given in metastatic melanoma, and we believe has an ideal profile of efficacy in terms of finding signal for efficacy of dual immune checkpoint blockade in rare tumors while minimizing as much as possible the risk of autoimmune toxicities, what we call immune-related adverse events. And in fact, in our study within the neuroendocrine cohort, the rate of high-grade colitis, which is one of the most common side effects with higher dose ipilimumab plus nivolumab, was only 6%, and we had no cases of pneumonitis or fatal immune-related adverse events in our cohort. We're not quite sure why the combination of ipilimumab plus nivolumab seemed to have a much higher rate of response in patients with high-grade neuroendocrine carcinoma relative to other studies that have investigated anti-PD-1 monotherapy in which response rates have been 0 to 5%. If tumor mutational burden is a pertinent factor and there's synergy between anti-CTLA-4 and anti-PD-1 directed approaches, that's one potential explanation, though this is something we're still investigating. At the current time, the standard of care for high-grade neuroendocrine carcinoma involves cytotoxic chemotherapy as the first-line regimen and as the second-line regimen. This unfortunately only has transient effects in which tumor shrinkage may last for weeks or months, and which long-term survival is limited, unfortunately, for patients with high-grade neuroendocrine carcinoma. Our hope is with validation of the cohort in dedicated high-grade neuroendocrine carcinoma within SWOG S1609 in an upcoming cohort that we can confirm these results and potentially lead to a new type of therapy, immunotherapy, with the potential for durable responses that last months to years, hopefully, in patients with this very aggressive disease. I think over the next 3 years, we're going to increasingly discover which rare tumor types are sensitive to immune checkpoint blockade. High-grade neuroendocrine carcinoma seems to be one of these types, so we're going to confirm this in a specific dedicated cohort of S1609 DART. It's very much the possibility that other rare tumors have sensitivity and efficacy for patients with these two agents, ipilimumab and nivolumab, as well as other immunotherapeutics, and we may discover that this is a type of therapy that has benefit above and beyond what we know in melanoma, non-small cell lung cancer, and the dozens of current common tumor types in which these agents have efficacy, that in fact several rare tumor subtypes may also have efficacy. And we have these as separate cohorts in S1609 DART that are currently accruing, and our hope is we'll learn more to benefit these patients with rare cancers who often don't have other options in terms of clinical trials or therapies for their disease. One of the unique features about SWOG S1609 DART is the fact that it's open at over 800 sites throughout the country. And in fact, for the neuroendocrine cohort, 2/3 of the patients came from community sites in which patients were close to home. And in fact, represents a viable option for patients who want to have therapy close to home and have access to the latest immunotherapy within the context of the SWOG and NCI studies that are available throughout the nation. We think this represents a success story for these types of trials and hope that more clinical trials are done for patients with rare cancers, especially close to their home.